And this was to sort of examine a typical average in metabarcoding data set. So the number of occurrences in the data set is on the x-axis, and the number of species is on the y-axis. And what you see is that most data sets sort of are accumulated in the lower left corner here, but the metabarcoding data sets, they actually both contribute a lot of occurrences and a lot of species to this picture. I've put iNaturalist and eBird in here for comparison. They are the biggest data sets we have. So I need to change this also. Yes, thanks for listening. Maybe I should say that. And then very shortly, some of you might heard it in the last presentation I did, we have been so lucky to get funding to work with improving this integration of eDNA data in GBIF. And it's, I won't go into the details here. Read the abstract of the other session and get in touch if you're interested in being involved in that work. Thank you for listening. And maybe I can't recommend to have two things to switch.
Creo que ya terminé la pregunta, pero te la repito. Con la llegada de la inteligencia artificial sabemos que el talento tiene que transformarse, ¿cierto? ¿Qué estrategias se están llevando a cabo para precisamente llevar esa transición?
Okay, if there is, I see I have 1 minute left, so if there is a question. Yes. Yeah, that's a really good question, but it depends in this case on how the person who shared the data chose to do it. So if they choose to cluster at 50% or something like that, they will be sharing a data set with 2 species in it. Or, but if they didn't cluster anything, it would be ASVs or something, and it would be, the unit would be more fine-grained than species. So it's on the data sharer to decide, but I would of course recommend to go for ASVs unless we know it's highly inflated or filled with errors in that particular type of data. Okay, thank you. And the next presentation is Sachit. Are you here? And I've chosen not to introduce people. If they want to introduce yourself, then you can do it better than I can. But welcome, Sachit. Take the scene.
This is advancing the real slides.
Thanks, Tobias. Good afternoon, Bogota. I'm Sajid Rajvandari from CSIRO Australia, and I'm also the part of Orbis Australia. Today, I'll be presenting Orbis Australia's efforts in standardizing and publishing fair marine eDNA data. And also, I'll be showcasing some of the work, collaborative work with our co-authors here. Okay. Sorry. Let me start with who we are. So Orbis is the Ocean Biodiversity Information Systems. It's a global open access database of marine species distribution. It's coordinated by the IOC of UNESCO. If you want to learn more about Orbis, we have an Orbis booth outside. Feel free to go there. I've extracted some of the stats from the Orbis website. Like we have published 165 million species observation. And also we have published 26 million DNA sequences. So I'll be focusing more on the DNA sequences here. In Orbis, we have 37 nodes worldwide. And Orbis Australia is one of the regional nodes. It is hosted by CSIRO NICME, National Collection and Marine Infrastructure at IDC Information and Data Center. So Orbis Australia has published till today 36 million records with 496 datasets. For DNA sequence, we have published 21 million and 26 eDNA datasets. So Orbis Australia also publishes some of the datasets from an Australian Antarctic program. So covering the Southern Ocean from 30° east to 155° east, and from Hobart down to the Australian Antarctic continent. Let's talk about what is eDNA observation, eDNA data. So environmental data, eDNA refers to the genetic material left in the environment. So how does eDNA data differs from the traditional observation data? So in the basis of detection, we see that the physical and visual presence is seen in the traditional observation data, whereas in eDNA, we use genetic traces in the environment. Other difference is species identification by morphology. Oh, sorry. Sorry, sorry about that. Thanks for reminding. Just let me know if I forget to click. Yeah. So species identification is by morphology, whereas in DNA, we use the DNA sequences. So we know that the process eDNA samples can be used to identify organism that identify or infer organism that has been in the proximity to the sample location across both space and time. So in this, we have different steps. We have sample collection, DNA extraction, DNA amplification, DNA sequencing and analyzing, detection, and data publication. But in this presentation, I'll be focusing more on the data publication side. As we talk about making eDNA data fair, so there are some challenges here. So existing vocabularies like Darwin Core and mixes, they only partially address the eDNA specific needs. And also there are some gaps in adopting these standards in the eDNA workflows. So to overcome these, a fair eDNA metadata checklist has been proposed. So this is the paper on this by Miwa Takasaki, and there's going to be a presentation from her in this session later on. You can learn more about it on that session. So what eDNA related standards, tools, and technology resources are available and what we are using. So for eDNA data publication, Orbis Australia is using Darwin Core standards together with the DNA-derived extension and also the mixes. And we are developing a pipeline for eDNA data publications. And for this, we are using other tools like Fairy tools and also the GBIF MDT tool. So what Tobias has presented earlier. And also the GBIF integrated publishing toolkit as well. And for anyone who wants to start publishing eDNA datasets, the best way to go with the eDNA publishing guidelines here is the QR code for the link, and also refer to the Orbis manual. So this is the very high, this is the very high level data pipeline that uses the tools like Fairy and MDT. So in this still, I've divided into the different sections. So data provider provides the data as a source. And once we provide the data source, the data is basically consists of the sampling and the sequencing data. So for the publication to Orbis and GBIF, we have to transform this dataset into the Darwin Core. Sorry. To the Darwin Core archive, and once we have the data in the Darwin Core archive, it goes into the Orbis, into the IPT server. And once we publish something to the IPT server, it gets harvested to the Orbis. And also, if that dataset hasn't been published to GBIF, we register them in the IPT server, and then it gets published to GBIF. So now I'll come back to our source data. Sometimes these providers already provide us the data in the Darwin Core format. So in that case, it's pretty simple. So we just go through the Darwin Core archive and do the publication. So in other cases, the second case could be like they provide the data sets, and we have a simple Darwin Core pipeline. We write the Darwin Core transformation script and produce the Darwin Core archive out of it. But now, as we have a different tools like a Fairy and MDT tool, so there are different ways. So let's go to the third way of doing. So we have a sampling and sequencing data sets. And there's a tool called Fairyator, which will generate a Fairy template. And we transform these data sets into the Fairy template. And once we have that data, we get the Fairy eDNA data. And Fairy also provides a verifying tool called Verifier, which verifies the data sets that we have. And then from there, we can do the Darwin code transformation and produce the Darwin code archive. And there's other ways. So we have a Fairy data, and then they have a tool called Fairy to MDT. So this transform the Fairy data into the MDT template format. And we use the MDT tool and produce the Darwin code archive out of it. And the last one would be like we have a sampling data sets, and we directly use the MDT template. And then we use the MDT conversion only mode, which will produce the Darwin code archive and then do the publication. So there are lots of possibilities. So we wanted to keep our pipelines possible to handle any kind of data sets in any formats that will be provided by the data providers. So here today I'll present the two use cases. The first one is the Australian Microbiome Initiatives. So here, Australian Microbiome Initiative, they didn't use the Fairy tool, but you make use of the MDT tool. So what Australian Microbiome uses the MDT tool. So MDT tool has an option to publish the datasets directly to the GBIF. And also they have an options like instead of publishing to GBIF, we can download the Darwin code archive out of it. So what we do is from the MDT tool, we get the Darwin code archive, and then we are working on the 3 different data sets. So 16 years bacteria, it has around 29 million occurrences. The Archaea one has 5.2 million, and then the 18 years has like 9.4 million records. So we push these data sets into the Darwin core, into the OBIS IPT server, and then the data gets published. Okay, so I'll just quickly go with the lesson learned from this. So for the first one is that publishing large, like I showed, like we have 29 million records. We are publishing the large data sets effectively through the IPT. So using the web interface of IPT, we couldn't upload that. The size of the Darwin code archive was around 29 gig. So we didn't upload it. So what we have done in this case is like we push the Darwin code archive directly into the storage folder of the IPT server. But there are other possibilities like we can split the files into different parts and then publish them to IPT. The other thing was leveraging the meta.xml files. So here you can see that the primary information, they are stored in the meta.xml instead of the DNA-derived extension file. So this saves a lot of size when we publish the data sets. So these informations are redundant for all the occurrences for that particular data set. So we could have, instead of publishing it in the file, we could put it in the meta.xml file. As I mentioned earlier, we use the conversion only mode for the GBIF MDT. And again, these data sets are pretty big. So using the web interface of GBIF MDT was pretty difficult. So there's an option that MDT provides the APIs. So all the data sets were published using the MDT's APIs. So it was pretty helpful and useful. And in this publishing the Australian Microbiome, we define themselves as the official publisher. Here we say when you publish, it's shown that the Australian Microbiome are the publisher instead of the OBIS Australia. The last point is data publication without taxonomic. So OMS is the taxonomic backbone for OBIS Australia. Sorry, OMS is the taxonomic backbone for OBIS. So in this case, like if no matches is found in OMS, then we'll have to go to the higher rank or even at the biota level. So if that dataset gets pushed to GBIF, then they can't make use of the GBIF taxonomic backbone, which is already available. So what we are doing is that we are not doing the matching for the taxonomic worms, matching to the worms at this stage. And that will happen at the OBIS. Once it gets harvested by OBIS, it will be done at that level. So, sorry. Another project that I talk is that the Global Marine Biodiversity Observation Global Partnerships. So CSIRO and Mindero Foundations are collaborating to advance the monitoring of Australian vast microecosystem. So in this way, they are in the 2 different phases. The first phase, we installed eDNA sampler in the RV investigator, and then the data sets gets automatically collected for each voyages in the RVI, and then the analysis and the discovery approach happens. So the next phase is like, we want to make this pretty generic so that installation can be happen for any kind of eDNA, any kind of SIP of opportunity, like any other SIPs, like the voyages can be used. So we also developed a pipeline for this. So I'll quickly go through the pipelines. We're running out of time. So in this, like, we are getting the two different sample data and the raw bioinformatic data sets. So we are using the Ferry tool for this. So after Ferry tools, we go to the Darwin code transformation, then Darwin code archive, and follow the same process. If you look at this, we are just skipping here the simple pipe, but the rest of the things are happening here. Few challenges that we face in publishing eDNA data sets. One is the taxonomic ambiguity due to the, I would say, incomplete reference databases. And other one is the dynamic taxonomy. So it means that the classification needs to be continually updated. And there's a complex data linkages connecting the eDNA sequences with the biodiversity sequences, which is pretty complex. So for this, maybe we need robust data pipelines and tools. Others is the Darwin code records become large due to the repeated information that I mentioned earlier. So for this, we could use maybe the new Darwin code data package could resolve these challenges. Sorry. For the last one, there are technical and limited barriers integrating the data from the sensors into eDNA workflows. So for this, like OBIS Australia is using the tools like GBIF, MDT tool, OBIS Pacman's pipeline, and also we are using some of the AI tools like the Chat IPT and other Chat GPT tools for the publications. So our current framework is we use the occurrence score and the DNA-derived extension. Then the next option would be like, as now there has been the addition of occurrence ID in the DNA-derived extensions, we could use event as a core, and then we can have with the event core model to better capture the sampling context and enhance the data integration and reuse. And finally, Darwin code data package is in the release. So with that release, we'll be able to link the event level information using the event assertion. And so, yeah, we are looking forward to the release of the Darwin code data package and how it goes. There are 77 different tables. It's complex, but I think they are working on with the specific case. And so we don't have to use all those 77 tables. We can use based on what kind of data sets that we are working on. That's my final slides. So why we need to publish the data sets. So here are a few of the points that we need to publish our data sets and what is needed. So the urgent need is like, we need local pipelines that is compatible with the local data systems that can easily be scaled as observation efforts expands. So for this, we are collaborating with the data providers. We are working with the GBIF. We are working with the OBIS for this purpose to develop generic pipelines that can be used for any kind of data sets and that can be scaled. Yeah. Thank you.
Thank you, Sajid. Yeah, we don't have time for questions. So the next speaker is Radasa from Sweden, talking about the Swedish ASV portal. Welcome. That wasn't an introduction. It was just
okay. Oh, wow. Just give me a second. I want to take a photo of all of you. It has been a long time ago, a talk in front of such an audience. So, hello, everybody. It's nice to see so many of you here and some friendly faces as well, which gives me extra pressure. My name is Rasa Bukontaita, and I am a coordinator for Swedish Biodiversity Data Infrastructure, or simple SBDI. I'm really honored to present one of our key tools today, the Swedish ASV portal, which is a platform for metabarcoding data, which I believe is a very important step forward to make metabarcoding data more accessible, open, and fair. Right. How does that work? Oh, okay. Right. Is it an easy way to make it work? Right. Is it there? I should point? Okay. Yeah, sorry. A little bit of technical issues here. But let me start with a brief introduction. What is SBDI? SBDI is Sweden's national infrastructure for open biodiversity data and tools. It's a consortium of 11 partners, as you can see, including universities and museums. We also host the GBIF Sweden node, which is managed by Veronica Johansson, which is also, let me read, the GBIF regional representative deputy for Europe and Central Asia. Did I get it right? Awesome. Who helps to bridge our national work to global biodiversity networks and communities. So this collaboration is very, very important for us. I'm not used to this. Right. SBDI mission is rooted into open science and open data, guided by FAIR and KIA principles. We also closely collaborating with Living Atlases and GBIF as well. It's really very odd to do this. We provide tools and resources that help researchers to synthesize, analyze, and share biodiversity data, which hopefully transforms biodiversity data into knowledge, scientific progress, and hopefully help with decisions for conservation and sustainability. Right. So if you would like to explore more of SBDI and our tools, so I would recommend to visit our website, biodiversitydata.sa. And now I would like to focus on my main topic today. So that is Swedish ASV portal, which is a platform for publishing, accessing, and analyzing metabarcoding sequence data. The development was led by Maria Prager and her fantastic collaborative team with many collaborators across SBDI and GBIF. The ASV portal provides a curated database, automated pipelines for data processing, such as denoising and annotating, submission support, helping users publish their data more easily and consistently. Right. So far, 21 datasets have already been published, which places Sweden among the countries with the highest number of published metabarcoding datasets. And this number is not only to be proud of. That is a very, very strong sign that researchers are engaged, they publishing, and they are sharing their data. So as a coordinator, let me briefly walk you through the workflow of ASV portal. So the users upload the raw sequences to, let's say, European Nucleotide Archive, INA. Then data is denoised and annotated. Once cleaned and annotated data is uploaded to ASV portal, which then is integrated into SBDI infrastructure. Then data is can be searched, it can be downloaded, can be analyzed directly from the portal, or can be explored in R using R package ASV Occur. So in short, I would say that ASV portal closes this loop ensuring that metabarcoding data remains accessible, reusable, and continuously improving. Let's try so for those who are more interested in technical aspects of ASV Portal, so ASV Portal is built on Docker Compose, which orchestrates different containers, which handles different components of the system, such as ASV storage, blast search, web interface, and also external connections with living atlases or GBIF. So I would like to focus on the three main highlights of ASV Portal, and that would be ASV storage and dynamic taxonomic annotation, blast search functionality, and our package ASV Occur. So then we're talking about ASV database. It's not just a simple storage. It's a living, evolving system. And what it makes it so special that annotations can be re-updated, or if you want, re-annotated as the reference database is becoming more and more available. Meaning that accuracy and resolution of taxonomic assignments improve without users needing to resubmit the data. It's automatic. So it's a fantastic, hands-off thing from the user perspective. Another powerful tool we have in ASV Portal is the blast search. You can search species sequences by species or family, genus, if you would like to, by primers, by target genes, and even by occurrences. As you can see, it's very intuitive and fast. It's very quick for users to filter the results and download what they need. And the last one would be our package ASV Occur, which was developed to make the downstream analysis more as smooth as possible, which allows to load, merge, or aggregate different data sets in our package. And one of my last slides, I would like to summarize that ASV Portal is a bridge between raw sequence data and global biodiversity systems, making it easy to reuse data, compare across different projects, and get new insights, new ideas. And if you have any questions, suggestions, ideas, please reach out to us. We are always happy to collaborate. More information about, again, SBDI and contacts you can find on biodiversitydata.sa. And my last slide, it's not this actually. My last slide is, yeah, where's my last slide, guys? Okay. There is no last slide. It's a dark screen, but what I want to say with the slide, the last minutes of my talk is I want to, yeah, here it is. I want to thank the Swedish ASV working group and collaborators for your dedication, for your experiences. Oh, all right. For this slide, you have a question. For your dedication and experience and making ASV Portal a dot on the global map. So thank you. And thank you all for being here and listening and for your interest in biodiversity. Thank you.
We have time for a question or two if anybody wants.
Uh-oh. If you have a technical question, I hope Tobias is going to help me to answer. Or two questions already.
Sorry, maybe this is technical, but when you have people doing all the bioinformatics on the website, it can be tricky, right? There's lots of decisions and
Yeah,
lots of things
yeah,
that can go
yeah.
wrong. Or
Yes.
do you have ways to walk people through that, or checks,
Yes,
or
yes, we do have submission help, and guidelines, and you can always contact us. So there is a lot of support out there because it's very tricky, as you said. It's also a lot of decisions to make.
Hey, Rosanna, really nice work. I have a question. Does this example from a Swedish institution could be applied or applicable to other, communities or other institutions? So essentially, this Swedish initiative could be an example of how to create like a local portal in which like local communities can share the Amplicon Sequence Variants. Thanks.
Thanks for the question. That's a great question. All the codes are open codes, so you always can, it's on GitHub, so just contact us and we can share all the information and codes. So you can definitely have it. And also, I don't know, do you want to mention something about GBIF and ASV portal? But you know more than I do. Yeah, then let's keep it a secret.
Yeah, I don't know, maybe my question touches on that connection between GBIF. But so
I'm joining the
what happens, so it's automatically re-annotated, but what then happens downstream?
What do you mean by downstream?
Well, where do you push the data to GBIF as well, right? So
Yes.
But so what happens with the re-annotated?
So it's automatically is updated. So there is a pipeline automatically updating it. If you want more information on how does it exactly happening, like through the codes and whatnot, please contact. Don't kill the messenger here. Thank you.
Yep, thank you. Next presentation is a recorded presentation. So I'm not sure if there's room for questions, and I'm also not sure if Miwa is online. She's not.
Hi everyone, my name is Miwa. I'm joining the conference remotely from Perth in Western Australia, the country of Wajak Noongar people. In the last two third-week conferences, I presented about the project to make eDNA data fair. This talk today will be a bit of an update about the project. So in the first part, I will briefly go over about the FAIR eDNA guidelines, and then I will get on to the main part, which is the title of this talk, the third week and GSC collaborative initiative to develop an eDNA metadata checklist. For a bit over 2 years, we've been working to develop the FAIR eDNA or FAIRY guidelines to make eDNA data fair. It's a multidisciplinary collaborative initiative with a lot of collaborators that you can see as the co-authors of the paper on the top right corner. This paper was published mid this year, and we also have the website available where pretty much all the journal paper contents are available, as well as any updates we made since the paper was published. The target audience of the guidelines are eDNA practitioners who are not necessarily familiar with the FAIR principles, data standards such as Darwin-Cohen mixes, or data publication procedures. So with that in mind, we provide a guide and tools for them to format their data in a standardized way with rich and machine-readable metadata, and then direct them to publish their data to databases such as GBIF, ORBIS, and INSDC. So I would like to quickly introduce the four main contents of the guideline. Firstly, the data components and the formats that you can see on the right-hand side. So we define required data components and standardized format. Then we developed a comprehensive metadata checklist. You can see the small fraction of it in the table on the left, as well as in the YAML format on the right. It's a very comprehensive checklist containing more than 300 terms, with 38 of them are being mandatory. And half of those 300 plus terms are from the existing data standards, such as Mixes, Darwin-Core, and DNA-derived extension. And the other half were created by the eDNA, FAIR eDNA working group. We then created a step-by-step guide for eDNA practitioners, starting with the template generations all the way to publishing data, then provide the published data link, such as DOI in the scientific paper data statement at the end. Where to publish data is described here on the right-hand side. Basically, we're recommending people to establish databases such as GBIF, ORBIS if it's a marine data, and INSDC for raw sequences, because this database can really make eDNA data fair with billions of other records that they host. The last part of this FAIRy guideline contents is the tools and the scripts. So we developed multiple scripts that can be used, that can be useful for eDNA practitioners as at these FAIRy practice steps. So for example, this is the top one here. It's a FAIRy ATA, FAIRy template generator. It's an R function to generate templates based on the study design. And then we have a verifier, the FAIR eDNA metadata verifier. Then also we have a FAIRy to MDT. MDT is a metabarcoding data toolkit developed by GBIF. So this kind of tools can reformat a FAIRy data into MDT ready format. We are still working to further develop more tools like this, and but I'm gonna cover more about it in a bit later in the slides. So these guidelines have been published. There have been several community uptakes, which is really exciting. So we've been running workshops for eDNA communities to introduce the guidelines. And now several eDNA labs around the world have implemented the FAIRy guidelines, the formats and a checklist in their workflows. So thousands of eDNA records that they have are now in the same format, and they're published with a rich metadata. We're also working with the relevant journals and the publishers to implement the FAIRy guidelines in their author guidelines. It just happened with the Environmental DNA Journal a few weeks ago, so that's really exciting, which you can see in the yellow highlighted part. Okay, so this was about the FAIRy guidelines, and now I would like to talk about our next step or current step, which is to develop the eDNA metadata checklist within GSC and TADWIC frameworks. The reason why this TADWIC GSC integration is important is because databases such as INSDC, GBIF, ORBIS ALA all use Mixes and Darwin-Core terms. So this collaboration and the integration will bring a direct link between the FAIRy to the databases. The first thing we did in this collaboration was to establish the eDNA task groups under these two organizations, TADWIC and GSC. So on the right-hand side, you can see the websites of these task groups with a list of team members. Members are in those two groups are the same because the TADWIC and GSC eDNA checklist will have a large overlap. So we are working together to develop both of them concurrently. And I would like to note that the TADWIC eDNA task group had actually been created a few years ago, well before the FAIRy project started, but it had been inactive for some time. So during the last year's TADWIC conference, we decided to reactivate it with the original members as well as the new members like myself and then other FAIRy members and other relevant initiatives. And as I mentioned earlier, The FAIRY checklist was built upon existing Darwin core and mixes terms, but we also created a new number of new terms that we as the eDNA community believe that they are important to include. So we're now reviewing the FAIRY checklist term by term to identify which of those new terms we would like to propose to TADWIC and GSC. So we're meeting fortnightly to progress through this review process. I would like to mention that when I say eDNA checklist within TADWIC framework, I mean that it's the Darwin core terms, including DNA-derived extensions and the terms, the proposed terms in the Darwin core data package tables, and also includes any FAIRY terms that are not in this Darwin core list, but we want to propose and hopefully get included. So we're working with the Darwin core DP team for that reason, and especially Cecilia from GBIF. We're reviewing the relevant Darwin core DP tables, such as molecular protocols and nucleotide analysis. And she's also helping us to review the FAIRY terms and with the eyes of Darwin core DP expert. So this collaboration has been really useful for us. So once we establish the eDNA metadata checklist under GSC and TADWIC frameworks, we will work on the mapping of FAIRY, Darwin core, and mixes terms, because we know that even though there will be huge overlaps between these checklists, they might not be identical. And so we will be using SCOS and SSOM, these type of mapping standards. And some of the Darwin core and mixes terms have already been mapped through the interoperability task group. So we will continue working on for the eDNA specific terms. We will then have these type of scripts available to prepare the FAIRY formatted eDNA datasets for data submissions. And these tools would convert the data formats and the term names to meet the data-specific requirements. And some of these tools are available already, and while others, like FAIRY to ENA, is still under development. We then want to implement the programmatic submission code to fully automate the whole process. And as a part of the FAIRY initiative, we recently started a specific working group for coding. So we will be focusing on this type of full automation with that working group. Thank you for listening to my talk. I would like to acknowledge all the collaborators for the FAIRY eDNA initiative, especially for those who are the members of the TADWIC GSC eDNA task groups, because they are putting a lot of effort to develop the eDNA checklist in this year. I would also like to thank for the TADWIC communities support funds that we received this year. So thanks to the funding, many of us, actually 9 of us from the task group could meet together in July in person and run workshops, meetings. We spent a whole week and we made some advancement with the task group objectives. So that was a really unique and significant opportunities, and we really appreciate that. So I would love to receive any questions, comments, and suggestions about the project from anyone in the room. And unfortunately, I'm not there at this moment. So please email me to connect or talk to many of the task group members who are actually attending the conference right now. So please talk to them and start up some discussion while you're there. And I would love to hear about it after you return your home. Thank you very much.
Okay, so we have a question from the public online. Is Miwa hearing us? Can she answer that?
She's connected. So
Okay.
see if we got her.
Okay, so there's a question saying, hi, your paper is very complete and cover all aspects of the data lifecycle. I would like to ask if you managed to transpose the checklist into real life. And we don't have an answer. Oh, okay, yeah, she answered. She said, yes, the FAIRY checklist has been implemented by several eDNA scientists labs currently. So, yeah. Any other questions from the public? No? Okay, thank you, Miwa, and thank you, everybody.
All right, I know how it works. Hello, my name is Catherine Silliman. I'm with Mississippi State University and the Northern Gulf Institute, working in collaboration with NOAA in the United States. And today I'm going to talk about some very collaborative work and effort being done to develop tools and a standards-based platform for DNA metabarcoding data, standardization, and publishing. So this graph is meant to look very complicated because it's really highlighting all of the different kinds of data and metadata that are generated when you're collecting, processing, and analyzing and publishing biodiversity data from environmental DNA samples. As you just heard in Miwa's talk, which is great, she went before me, there's now been a lot of effort to develop standards to tackle this very complex data landscape, an increasing number of tools, which is very exciting. And so our group has also been contributing a number of tools, leveraging these FAIR-E data standards in order to help make eDNA data more fair. You can take a screenshot of this now or a picture. I'll throw it up again at the end. But this is going to be a real high-level, quick overview of some of the methods we've been developing to support our data, but they're also, I think, really broadly useful to anyone working with eDNA data. We have FAIR-E sheets, which will take that FAIR-E metadata checklist and create Google Sheets for you to input your sample collection data, analysis data, processing data. We have an eDNA data publishing workflow called eDNA to OBIS, which again is leveraging data formatted using FAIR-E, and we'll create a Darwin Core archive. We have FAIR-E to NCBI, which will create the files you need for publishing raw sequence data to NCBI, biosample, and SRA. And then we have a new database and web portal called the Ocean DNA Explorer. I'll be showing some great demos from that in just a little bit. And so FAIR-E sheets, everything really starts with this. This is a very easy to use pipeline to generate one of these templates in Google Sheets. You can configure it to make the template work for your specific project and the kind of data you're collecting. And it will create each of the sheets you need for project metadata, sample metadata, experiment run metadata. So if you're starting from the very beginning of a project, you can already be collecting data using these community standards. And then once you get it in that format and you analyze your data, which is definitely probably the hardest and most time-consuming part of all this, now you're in the one format you need for publishing your data on the Ocean DNA Explorer, for converting it into the files you'll need for NCBI and OBIS and GBIF. So the eDNA to OBIS pipeline, if you were at Tadwig 2 years ago, I presented the first iteration of this pipeline. Since then, Baden Willems, a software engineer in our group who's here today as well, has turned it from my Jupyter notebook into something that's automated, really easy to use, and with a lot more increased functionality. And so it will, transform your data from that FAIR eDNA standard into the Darwin Core standard. But also notably, it will align whatever taxonomy you put on your sequences from any database, and they all use their own taxonomic backbones, it feels like, and it will align them to either the worms taxonomic backbone, which is what OBIS prefers, or to the GBIF taxonomic backbone. And then it will also accommodate multi-marker data sets. And so ourselves and our collaborators at NOAA, a lot of the surveys we do, we're running lots of different markers and assays on one environmental sample in order to get a wide range of different taxonomic groups. We want to really clearly link the hierarchy of those different data sets to the original sample they came from. And so you can create one Darwin Core archive from multiple markers, but also maintain enough information to easily separate those out back into the original markers data sets they came from if you needed to for downstream analysis. So as I said, everything starts with getting your data into FAIRY sheets. And the output will be a DNA-derived extension file, an occurrence core file, and then a really great taxonomic assignment info. So we'll give you some information about how the taxonomy you gave your data maps to the backbone you chose. And it can help you identify the cases where that mapping got a little wonky, which always happens. And you can catch that before you publish it on GBIF or OBIS. It also optionally will generate an extended measurement or fact. So if you have temperature, salinity, other kinds of data, you can share it that way. And optionally an EML file for project metadata. It comes with a really great HTML report. And this is just showing that it's just one quick command to run. It's very easy to run. And you customize it using a number of configuration files, which are really excellently documented on the website. This is an example of a successful report, but we'll also give you some warnings like, hey, you don't have any times with event date. Are you sure that's right? Hey, you don't have geodetic datum. That's really recommended. Or do you want to include that? And so it gives you some good feedback there. And with the time I have left, I want to highlight the Ocean DNA Explorer. And so this is a platform for data publishing, visualization, and searching for marine metabarcoding data. From the homepage, we have links that will take you out to explore the projects we have on there, the samples, the sequences, the taxonomy. What I do want to highlight is the site is live, the data is real, it's very functional, we want you to go on there, but it is still in active development. And so we would love for folks to try it out, to interact with it. And we provide links for you to report the bugs you find, to make feature requests, and help make it a better resource. And so why do we need another database and web portal for eDNA data? We found with our collaborators at NOAA the need for something that was really focused on the applications that NOAA cares about: biodiversity monitoring, ocean exploration, fisheries, protected species. It is also all open source. So the database schema and the website code is on GitHub, which really helps with reproducibility and can be a model for other people who are looking to make maybe similar databases, web portals as well. It really gives us the flexibility to develop very advanced search and API capabilities. You can make queries and synthesize data across all of the eDNA data sets, either through the web portal itself, and I'll show a demo of that in a second, or programmatically with code. And because it's built on the FAIR eMetadata standards, it leads to this interoperability between global sequence and biodiversity repositories. So we'll see if the video works. This is a demo that Baden put together.
Let's learn how to find data across OTUs.
Make it a little louder.
Explore page lets you view any row of any table in the database. Then you can view more information by clicking on the row.
Can you
Select
make the
which
video
table to
louder?
look at using the tabs here. And
So
you
here
can
he's
customize
exploring
what
the samples. You can do lots of different great filtering based on the metadata that you have. And then this helps you look at all of the different kinds of tables that we have in the database. For your samples, your assays, your metadata, you can search, give me all of the data that's available in a specific region. It's got, good text search capabilities. But if you wanted to do more complex searches, we have an advanced search feature, which will help you integrate across the different kinds of table and data that we have on the database. So really take advantage of the relational structure that we have for the
NOAA projects.
how they do beta.
Analyze 16S assay.
And it's got great functionality. And we would love for you to go and play around with it. And then you can download the data that you find using this search feature. This is just in case the video didn't work, which it did. Also, I'll throw up the link again, but you guys can check it out right now on your phones. It's mobile friendly, which is great. And if you're more programmatically inclined, it has a really robust API and well-documented API. And so you can, with just a few lines of code, download and search the database for the data you need and really do some great synthesis across the different data sets and the different markers that we have on there. And so this is really, I think, where the power of having this database is going to come out. Uploads right now are invite only. We're focusing on NOAA collected and NOAA supported data. But if you feel like your data would do well on a portal like this, if you're interested in collaborating, definitely please reach out to us. We would love to hear from you. Please keep an eye on this space. In the future, we're going to continue with our feature improvements. And then the ultimate goal is to incorporate an automated eDNA to OBIS publishing pipeline on the back end. So you can submit your data to ODE, and it will do all of that processing into the Darwin Core archive for you. And so it's just more of a one-step publishing process. We want more data sets, more users, and we really want user feedback from the community. So please do go and check it out. And then if you have any questions, come find us. There's myself, Baden Willems, our software engineers here, Luke Thompson, who's the lead of our research group, and we'll be giving a talk Thursday afternoon during the MBON session. And with that, I'll take any questions.
Really nice presentation, Catherine. Thank you very much. And also, well, thanks for sharing these tools. I'm really keen on taking a look at them and learn more about how they could be applied in our communities. I have a question for the FAIR e-sheets in particular. As I understand, then you have these Google spreadsheets that can be shared and filled in by researchers. I wonder if they also incorporate some like control vocabularies or ontologies, so then that facilitates that they actually start adopting, the machine actionable kind of, control vocabularies and semantics. Thank you.
That was a planted question. No, not really. Yes, it does. So we have some drop down, we have some data validation that it comes with. So it helps you use the controlled vocabularies from the get-go and flag issues. Yeah. Thank you.
Hi, thank you very much for the great presentation. I was wondering, because all of the data is processed, right? Does the metadata get leveraged in the process in some way, or what are the sort of like algorithms that are being developed for processing the data? Maybe in light of the metadata, but I don't know.
So we have also developed an Amplicon sequence processing pipeline that does not explicitly use the metadata, but it will take metadata to help with some summary output statistics, for example. eDNA to OBIS will leverage some of the metadata you're providing, certainly for making like the project metadata. But in terms of like how it actually processes, yeah, I don't know.
Okay.
Yeah, maybe, yeah. Talk to me later. Yeah, maybe I didn't quite get the question. Okay, thank you.
Thank you. The next presentation is a recorded presentation as well. And after that, we have a break, but we continue after the break here again with the 5 last presentations of this session.
Hello, everyone. Thank you so much for joining this session. My name is Camila, and I'm from Global Biodiversity, which is a research center focused in biodiversity and genetic resources based in the north of Portugal. So today I'm going to be presenting the progress that our task force has achieved by proposing interoperability improvements in fairy DNA data by aligning metadata standards. And this work is currently being developed under the European Horizon funded project, eDNA Plan, and has been carried out by me and other task members that you can see listed here. So that's Emily, Joanna, Peter, Sarah, and Vera. So several analyses have now reported that current metadata practices in eDNA data sharing do not uphold fair data principles. So this means that data isn't always findable, accessible, interoperable, or reusable. And in this presentation, I'm going to focus more specifically in interoperable principle. And interoperable just means that data can be easily shared and used across different platforms. The fact is that in reality, metadata is often fragmented. So this means that metadata is stored in different places across the data sharing workflow. It is also diverse, meaning that because there are so many different standards, similar use cases can use different standards. And this fragmentation and lack of consistency makes it very hard to reuse data. It limits interoperability, and ultimately it just slows down large-scale analysis and scientific discovery. However, before diving into it, I just wanted to share the outcomes that have been produced within the project and have already been shared or will be shared in this conference. So if you want to read more about other deliverables, you can search for us in Zenodo. And within eDNA Plan, the consortium shared earlier today proposals for improved fairness in the eDNA space and collaborations within official data standard bodies for better metadata reporting. And these are marked in green because they already happened. But tomorrow, our colleague Katrina Axter in the poster session will share a poster on our vision for the future eDNA digital landscape. So please stay tuned for that. Our main goal in this task is to develop models that align existing metadata standards. And with this, we aim to build foundations so that interoperability in eDNA data can be improved in the future. And why are these needed? So standards can be misaligned. This means that they are not tailored to a specific field, and in this case, they are not tailored specifically to eDNA needs. We can also have inconsistent reporting where users just interpret the same metadata terms differently. And for example, we can see here below an example of this. For example, we could have 3 different users using 3 different standards to report eDNA data, where all 3 of them have very similar terms, which may or may not relate to the same study characteristics. Users may also choose the wrong checklist for the data, and this all just results in increased interoperability challenges where moving data across different platforms becomes more difficult. I also wanted to bring you an example of how the exact same terms can have different representations. So here we can see the expected values for specific hosts for the Darwin Archive DNA-derived data extension and the mixed score standard, which is minimum information about any sequence, can have the same terms, however, expect different values. For the DNA-derived extension, you can see that it only expects the tax ID. However, for the mixed score, you can see that it expects the species nomenclature and then a pipe and then the tax ID. And now you must be wondering what is specifically an alignment model. And I'm going to be a little bit more technical before providing just a more visual example. But alignment models are a systematic method to map terms between standards. And a mapping describes how a subject is connected to an object by defining the nature of their association. And if it does, in fact, look a little bit ambiguous, let's just have a look of how this can be played out. So here we have a visual schema of a mapping between 2 ontologies. And this is using the SSSOM approach. So you can see in the sources here below the image that SSOM stands for Simple Standard for Sharing Ontological Mappings. So if you look closely into it, we can see that there are two terms being mapped between two different ontologies. So on the left in pink, we have ontology A, which is EFO, that's the experimental factor ontology, where the term or subject is adenofibroma. And this is the term that we're mapping from. On the right in red, we have ontology B, which is the mondo. You can see the different standards, the ontologies in this case, under the subject ID. So mondo is the mondo disease ontology, where the object is also adenofibroma. And this is the term we're mapping to. In the middle, you can see the predicate. So the predicate describes the type of relationships between these two terms. In this case, it tells us that these terms are an exact match, which just means that their definition is essentially identical. And below, you have the mapping metadata. So this is information about the mapping process itself. It tells us, for example, that the mapping was done on the CERN label. So this means that the mapping was done on the CERN name. It was done in May 2021. It was human created by a researcher with this RCID, and it was also done with the help of a tool called the RFD Matcher. So what's the purpose here? The purpose is to compare standards, and this allows us to bridge gaps between them. It is also to align the term meaning, which is semantics, and the term usage, which is syntax. And I know these terms are confusing, and I'm going to go over them a bit throughout the presentation. So I think it's worthwhile to just have a few examples to consolidate this. So semantic mismatches occur when the meaning of the term does not align. For example, we can have the term habitat, and this usually just refers to an environment which is specific to a species or to a community. And this is different from an environment type, which usually just covers a broad geographical area. On the other hand, we can also have syntactic mismatches. These happen when the format of the data differs. For example, one standard might record the temperature in Celsius, another one in Kelvin, another one in Fahrenheit. And the fact is that these values are in fact different, but technically they are the same. So when we take these alignment models into consideration, we can greatly improve interoperability. And how does this work? So by using information in these mappings to create automated data conversions, we can translate the metadata between the standards and make different platforms that use different standards work together smoothly. So this allows data from multiple sources to be easily shared, to be easily combined, and to be easily reused. And I wanted to bring a little fun example. So for example, on top of the slide, if we consider the standards as bugs and or mappings, which is the information between the relationships of these terms, as web connecting the standards, the conversion workflows or the conversion tools would be the spider that weaves these links and builds the translations. So our work has been is built on previous efforts to align the Darwin Core and the Mixed Core standards. And this effort aimed to build sustainable interoperability similar to us, but then their focus more on the main standards that were used in the OMIC biodiversity data. And in case the standards aren't familiar to you, I'm just going to briefly walk you through the eDNA data landscape that is relevant to this presentation. If you want to explore the full picture later, you can go into the Zenodo and search for our deliverable 15, which we have this whole landscape on the eDNA space. But for now, I'm just going to focus on the eDNA sequencing data and occurrence data. So on the top right of the image, you have eDNA sequencing data, and this is uploaded to databases that compose the International Nucleotide Sequence Database Collaboration. And this includes the European Nucleotide Archive, which is based in Europe, the National Center for Biotechnology Information, based in the US, and the DNA Data Bank of Japan, based in Japan. So all of these use mix, and that's the minimum information about any sequence to report sequencing data. On the other hand, occurrence data, which provides evidence that a particular taxon was found at a certain place at a certain date, is uploaded to various other platforms. So this is the left lower corner of the image. So I'm just going to briefly go over the main ones, which are GBIF, that's the Global Biodiversity Information Facility, and OBIS, which is the Ocean Biodiversity Information System. These use the Darwin Core Archive. And excitingly, and more recently, a consortium led by Miwa Takahashi proposed the FAIRY checklist, which is a new checklist designed specifically for eDNA needs. And it's been getting traction within the community, and it's now being further developed in collaboration with standard bodies. So we hope that soon it's going to become an official eDNA extension. So here's a quick overview of our approach that we use to align the DNA-derived data extension and the mixed score standard. So we followed 5 main steps. So first we did a review of all the DNA-derived data extension terms, and then we established the associations between those terms and the terms in the mixed score. So we mapped them. And we used the DNA-derived data extension version 1 and the mixed score version 6 using the SSSOA framework that we previously saw. So next, we identified any gaps or inconsistencies and we documented the provenance, mapping justifications, and confidence levels for each mapping. We're currently in step 5, so we're carrying out validation and we are compiling feedback to refine our results. And I wanted to note that all these 5 steps, mainly the mapping terms, are done twice. So one is for the semantic alignment. So this means, once again, that we check whether the meaning of the terms match. Another one is for the syntactic alignment, which ensures that the formats or data structures are compatible. And this approach was established under the guidance of Pierre-Luigi Bordaggi, which is a member of the eDNA Plan International Advisory Board. And after completing our analysis, we identified several key findings and gaps. So first, we found multiple version mismatches. Oh, I skipped a few. Yeah, okay. So after completing our analysis, we identified several key findings and gaps. So first, we found multiple version mismatches across the terms. So specifically, terms in the eDNA-derived data extension that were based on the mixed score expected different syntax values. So I brought here a more visual example. So here we have the taxonomic identity marker term, which is taxident. And for the DNA-derived data extension version 1, it required the same formatting as the mixed score version 5. However, with the update of the mixed score to version 6, this consistency no longer holds. Now, if a study does not use 16S or a multi-marker approach, this field under the mixed score version 6 will need to be filled with other, simply other. However, when you use the DNA-derived data extension for the same term, because it still has not been updated, you will need to fill other semicolon and the specific taxonomic identity marker you use. So this just highlights a gap in alignment between the mixed score and the DNA-derived extension, which does impact standardization across studies. And it's also important to note that the mix is also adding for another update, moving to version 7 soon, which can also just amplify these alignment issues. Another gap that we identified was missing provenance. So provenance refers to the origin of a term, how it was created, why it was created, the rationale behind it, and the context supporting these decisions. And for terms in the DNA-derived data extension, so here you can see the figure on the left. And these terms were based on the MIC checklist, and that's the figure on the right. And MIC checklist just stands for minimum information for publication of quantitative real-time PCR experiments. So for these terms, we were unable to find the information of how they were derived or their definitions were established and the reasoning behind them. This makes it very difficult for us to just confidently determine accurate mappings. So based on this, we identified critical requirements that are needed to take into consideration for mapping efforts. So first, mapping provenance is essential. We need to know who created a mapping, when it was created, which version was used, and why it was created in that way. We also believe that it's important to add reliability scores that show how confident you are in the mappings that you have established. And finally, term relationships must be considered. So I haven't gone over this, but terms rarely match on one-to-one across standards, and they are often hierarchical. And just for an example, you can see that, for example, animals and mammals are not in the same hierarchical level. So animals is a broader concept, while mammals is a narrower concept. And these relationships are quite important to consider in your mapping effort. So these requirements make transparency essential in any mapping efforts. Users need to understand the limitations and the reliability of mappings to make informed decisions when using this information and when building interoperable systems. And the transparency can be achieved, for example, by reporting mapping accuracy, so including known errors, confidence levels, and integrating community feedback. And it can also involve reporting the completeness of the mapping. So for example, does the alignment cover all terms of a standard? Which terms are missing? And are there any limitations in the scope that you considered for the mapping that you are doing? So currently, as I mentioned previously, we are awaiting feedback on our mappings. And our next steps is to continue and expand our work. So I plan to extend the mapping efforts to the FAIRY checklist that I mentioned briefly before, and other environmental and measurement extensions commonly used in the eDNA research community. And additionally, also aim to share our work in platforms that facilitate communication in the community and feedback. Our work is in GitHub. It's still private, but we hold for the feedback of our official standards, but we hope to share with the broader community. So this approach allows us to achieve long-term impact. So by collaborating with official standards, such as the Taxonomic Databases Working Group and the Genomic Standards Consortium, we can develop work that was previously, under previously established memorandums of understanding. These are documents that define how these institutions communicate and how they can work together to advance standards. And by working under this, we can guarantee that our work will be considered when they upload their standards. And also by fostering community ownership, we hope that our work can also serve as a foundation for future mapping efforts. So as main takeaways are that alignment models provide a strong foundation to improve interoperability between platforms, and they do support fair data sharing principles. We are also formalizing this process by mapping standards adopted by the eDNA community and ensuring continued sustainability. And that's a wrap. Thank you so much for attending this presentation. You can stay up to date with our work by following our project socials. You can just search for eDNA Plan on major platforms. And we do hope that you're inspired to get involved in shaping the future of the eDNA digital ecosystem. Thank you.
That was exactly 15 minutes. See you after the break. When is that? Is that half an hour or what is it? Half an hour, if you want to continue here. Have a nice coffee break.
President Murphy. No,
Get it fast, if it's fast enough so we can fly away, you gotta make a decision. Leave tonight or live and die this way. So I remember when we were driving in your car, speed so fast I felt like I was drunk. City lights stay out before us, and your arm felt nice wrapped around my shoulder, and I had a feeling that I belong. I had a feeling I could be someone, be someone, be someone. Got a fast car. We go cruise and entertain ourselves. Still ain't got a job. Now work in the market as a checkout girl. I know things will get better. You'll find work and I'll get promoted. And we'll move out of the shelter. Buy a bigger house and live in the suburbs. I remember when we were driving in your car. Speed so fast, I felt like I was drunk. City lights lay out before us. Your arm felt nice wrapped around my shoulder, and I had a feeling that I belong. I had a feeling I could be someone, be someone, be someone.